So what you're currently looking right now are two pairs of twins, right? So twin pair number one, twin pair number two. So they both married to the other one in the twin. So this is a couple, and this is another pair of couple. My question for you is, if two pairs of identical twins married each other, would their children be identical to each other? In this video, we're not going to review the answers, but I'd like you to think a little bit deeper in terms of their genetics composition. So, in this video, we're going to study what makes twins identical, right? Twins are identical because they have the same composition of nucleic acid. So nucleic acids are the instructions or blueprints of life that tell the life what needs to be made. So DNA and RNAs are the two common type of nucleic acids. DNA contains a unique blueprint to make protein. That protein would actually determine the trait, such as whether your earlobe is hanging or if you do not have a very specified earlobe. So let's dive deeper into nucleic acid. Nucleic acids are organic compounds made of the monomer nucleotide. And what you see on the right side is the molecular structure of one nucleotide. You can see that it is made of the following elements. We have carbon, which is represented by the turning points here, carbon. And also there's a metal group, right? There's a carbon with two hydrogens here. So we have hydrogen also. Notice that we also have oxygen, phosphorus, and nitrogen. You can see that nucleotides can be divided into three parts that makes one nucleotide. So the orange circle here, that's a phosphate group because it contains one phosphorus and four oxygen. The blue group over here is representing the sugar, right? You can notice that this is a ring-shaped structure of a glucose, and it has 1, 2, 3, 4, and 5 carbon. So that's why it's also known as a pentose sugar. Pent represents 5, so 5 carbon sugar. Next, we notice that this yellow group right here is containing nitrogen, so it's also called a nitrogenous base. Nucleic acids, the more common one is DNA, but the lesser known among yourselves is probably RNA, right? But since COVID, most people also know about RNA as well. So we have DNA and RNA, both are examples of nucleic acids. You're expected to know the full name of DNA and RNA. DNA stands for deoxyribonucleic acid. And RNA stands for ribonucleic acid. You'll notice that they have very similar composition, right? They both have phosphate group, right? DNA both and RNA both have phosphate group. They both have bases as well, but the composition of their base is slightly different. And they both have the sugar, but the name is slightly different. So let's look at their nitrogenous base first. So nitrogenous base, as we said just now, is one of the parts of a nucleotide. So for DNA, you have two types of nitrogenous base. First type is what we call a pyrimidine. So pyrimidine is containing cytosine, thymine, and uracil. DNA contains the nitrogenous base called thymine, whereas RNA only contains the RNA. Instead of having thymine, it will have uracil as a replacement. But notice that among cytosine, thymine, and uracil, they only contain a single ring for its nitrogenous base. The second type of nitrogenous base is what we call purine. So instead of having one ring, it will have two rings instead. One ring, two ring, one, two. And they are adenine and guanine. For sugar, DNA and RNA also contains different sugar. For DNA, it contains deoxyribose. That's how DNA gets its name. And if you remember, O's means sugar. Whereas in RNA, the sugar is ribose, R-I-B-O-S-E. So if I have a nucleotide and I want to connect one nucleotide with another nucleotide, notice that it will have two hydroxyl group that they can form a reaction together, right? So we use condensation reaction or as what we have learned, dehydration synthesis. Which we take one H and one OH from the nucleotide. As a result, they're going to form a bond between that two nucleotides while taking away one water molecule. That bond that you just formed between the sugar of one nucleotide, so remember this is the sugar part, the pentose sugar of the nucleotide, and this is the phosphate group of the other nucleotide. That bond is called a phosphodiester bond. So you can see that nucleotide can be linked together by a phosphodiester bond to build a dinucleotide. If you repeat this process, it will form polynucleotide. Remember, di means two, poly means many. A phosphodiester linkage consists of a phosphate group that links to the sugar of the two nucleotides. This links will create a backbone that consists of the sugar and phosphate. Hence, we call this sugar phosphate backbone. So here is a picture of the sugar phosphate backbone of 4 nucleotides. You can see that we have one nucleotide that consists of phosphate group, sugar, and the nitrogenous base, and you have another nucleotide. There's another nucleotide, and there's another nucleotide. Notice that the phosphodiester bond will connect the phosphate group and the sugar, right? You can form a chain of that backbone. So that's what we call a phosphate sugar backbone. And it has numbers that indicate starts and finish. 5 prime represents the starting of the phosphate sugar backbone, and 3 prime represents, all right, you notice that with 3 with an apostrophe at the top, that is 3 prime, so that's the end of the phosphate sugar backbone. So the nitrogenous base can actually form bonds as well, but they don't form bonds with the phosphate or the sugar. Instead, it will form bonds with the other base of other nucleotides. So in the case of DNA, you can actually form bonds, the hydrogen bond, between bases of 2 separate strands of DNA. Notice that if they do form bonds between 2 strands of DNA, the 2 strands are going to run in opposite direction. On the left side, you have 5 prime running to 3 prime from top to bottom. But on the opposite side, you have a 5 prime to 3 prime running in opposite direction. So that's what we call a double helix, right? Double helix means you have 2 polynucleotides spiraling that forms this particular unique shape called double helix. The backbone will always run in the direction of 5 prime to 3 prime. The 2 strands are going to run in opposite direction, and we call this particular shape anti-parallel. Anti means in opposite direction. Parallel means that they're going to run in parallel lines, right? It's kind of like two lines that runs parallel to each other. The way how they pair up also has a specific rule. Notice that C will always pairs with G. C represents cytosine, G represents guanine. D, thymine, will actually always pair up with adenine. The type of bonds that forms between A, T, G, and C's are hydrogen bond. T always pairs with thymine, A always pairs with T, thymine. G, guanine, always pairs with cytosine. You also notice that the number of bonds is slightly different. For CG pairs, you always have 3 hydrogen bonds. So I'm gonna write here, CG pairs always have 3 hydrogen bonds, whereas A will have 2 hydrogen bonds with T. This particular pairing rules is called complementary base pairing. So just remember, complementary base pairing means A pairs with T, C pairs with G. If you remember, in previous slide, we also mentioned that in RNA, T is being replaced by U. So A can also pair up with U if it's in the RNA case. So let's look at RNA. RNA doesn't always form double helix, but it can actually form hydrogen bonds as well within itself. So notice that this is an example of RNA, and we know that this is an RNA because it has U instead of T. So notice that it can form a shape just like a protein being twisting around and start folding within itself. And there's going to be a hydrogen bond formation between the bases within the RNA. So RNA is mostly single-stranded. I'm gonna add the word here, mostly. We do have double-stranded RNA, but it's just not very common. So the rule of complementary base pairing will also occur between the bases in RNA, or between RNA and DNA, or between RNA and RNA. So it can form between RNA and RNA, or RNA and DNA. Remember, in RNA, you do not have T, you have U, which stands for uracil. So as a result, A will pair with U instead. It will also form 2 hydrogen bonds. So one thing that we need to make clear in this video is a particular molecule that we also see very often in biology, but not many people are aware of this. And this is ATP. Notice that it has 1, 2, and 3 phosphate group. Connecting to a ribose sugar and a nitrogenous base, which is adenine. So ATP stands for adenosine triphosphate, right? Commonly known as ATP. This is a very important nucleic acid that we use because it serves as the energy currency of the cells. It means this is providing energy usage for a lot of the things that happens within the cell. And ATP is produced during cellular respiration, which is the process of extracting energy from food. So if we have food, that energy from food is being extracted and it will be stored into the form of ATP from ADP. So you can think of ADP as being converted into ATP by adding this 3rd phosphate group here is in order to store energy. As a result, now ATP can be used to power up your cell. As the cell uses the energy from ATP, that P will be released, then it will become ADP again. ADP stands for adenosine diphosphate, which means 2 phosphate group only. It has a lower level of energy. Then it can be recharged again if you have energy from food. So this is also what we call ATP cycle. So remember, ATP is also a form of nucleic acid. So to summarize it, nucleic acids are the instructions or blueprints of life. Nucleic acids consist of carbon, hydrogen, oxygen, nitrogen, phosphorus. The monomer is nucleotide, which has phosphate group, pentyl sugar, and nitrogenous base. It can be categorized into DNA or RNA, differ in the structure and function. ATP is also another example of nucleic acids. With that, we end the video, and I will see you in the next video. Bye-bye.